Hello everyone, this is lecture 2 in BE544. In the previous lecture, we mentioned that there is something called digital histopathology, which is used in the archiving process to prepare the specimen. So what is digital histopathology? Digital histopathology refers to the digitization of traditional histopathology slides. This will allow the pathologists to view and analyze tissue samples on a computer screen rather than using a microscope every time. This process involves scanning glass slides. These glass slides will contain tissue samples. The scanner or digital histopathology will contain high-resolution images. The major advantage of these digital images is that they can be stored, shared, and analyzed using any specialized software. So instead of using a microscope every time, you can access using a computer these digital slides every time. You can apply annotation on it, as you see here in the red color. Extract these regions and apply any processing after that. You can also include these digital slides to any system, so you can use it in any classification system that can accept the slide and predict if the slide is benign or malignant, if you are talking about a cancer or something. And we are going to go in depth in the classification process in the next couple of lectures. So we mentioned you need a scanner to scan the slides to make it digitized. So you need a scanner. So scanners play a crucial role in converting traditional glass slides into digital images. These scanners are specialized devices designed to capture high-resolution images of histopathology slides with precision and accuracy. And it depends for sure on the scanner itself and the mechanism of using it. You apply the slides and you insert it into the scanner, and then the scanner starts to process each of them. Each scanner have different parameters or configurations. So scanning parameters are critical settings that determine the quality and the characteristics of digital images. The first thing is magnification. So magnification here specifies the level of detailed capture in the digital image. It can range from low magnification for an overall view of the tissue architecture to high magnification to get a detailed examination of cellular structures. It can be 10x, 40x, or 400x. For example, as you see here, you are getting an overview of the tissue architecture, but when you apply high magnification from 20x, 40x, and 100x, you are getting more details. So for example, assume this is a nuclei. If you went back to 10x, you will not find that nuclei or the nuclei in that image. Another example, here 10x, so you are getting an overall view, and through increasing the magnification level, you are getting 20x, 40x, 100x, so you are getting closer and closer to be more detailed. A second parameter is autofocus. It ensures that the scanner maintains sharp focus through the scanning process, even when scanning large mounted slides. The third one is stitching. It determines how multiple image styles are seamlessly stitched together to create a complete digital representation of the slide. However, the major one or the major parameter here is the magnification level, and we are going to deal with different magnification levels, extract tiles from it, and we are going to understand what is the meaning of a tile or a patch and how to extract it, how to process it, and how to apply it to a classifier or something. So you have a physical slide, you applied it to a scanner, and then you get a digital image. This digital image called whole slide image. So a whole slide image or whole slide scan or virtual slide, it refers to a high resolution digital representation of an entire histopathology slide. So by applying this whole slide image or the digital image to a software, you can navigate through the whole slide image at any time. So unlike traditional microscopy, you can only view a small portion of the slide, while whole slide image captures the entire specimen at once. The second thing, the whole slide image preserves spatial context and enable comprehensive examination. So as you see here in this figure, you are getting a whole slide image. If you apply this digital image to a software, you can zoom in and zoom out to get fine or more details. So if you are a pathologist, you can say, yes, this is a cancer region, this is a benign region, and so on. So assume now the scanner scans the whole slide image at 3 different levels, for example 1x, 10x, and 40x, and you need to organize, or the software needs to organize this hierarchical structure. There are multiple ways that the software can handle it or the scanner handle it. The first one is pyramidal view. It organizes the whole slide image into a hierarchical structure, as you see in this figure. It makes it like levels, so the top level is the lowest resolution, and when you get deeper and deeper, you are getting more undefined details. So if you are targeting an abstract view of the whole slide image, you can present the top one, while if you are going to process or you are going to work on the fine details or the detailed structure of the nuclei or the cytoplasm and so on, you can navigate to higher magnification levels such as 10x or 40x. So this organization will conserve the computational resources by loading only the necessary image data based on the user's viewing area and zoom level. This will enhance the performance and efficiency in image analysis. The second way is tiled view. The tiled view will divide the whole slide image into smaller regions. These regions are called tiles or patches, as you see in this figure. Each tile or patch will represent a portion of the overall image. Based on the software, these tiles are loaded and displayed dynamically as the user navigates through the image. It will allow an efficient streaming of image data, and it can be very useful on devices with limited memory or bandwidth. So as you see in the grid here, you have a whole slide image split into regions, where each region represents a portion of the overall image. The third one is the multi-resolution representation. It will involve storing the whole slide image at multiple resolution or levels of detail. Instead of organizing the image into a pyramidal structure, each level of detail is stored separately. So you can store the first level in a file, the second level in another file, and so on. This will allow the user to switch between different resolutions as needed. This is a flexible approach in accessing and analyzing image data, but it for sure will require more storage compared to the pyramidal view. As we go on, we will be focusing more on the pyramidal view, which is considered the most common used approach in handling and organizing the whole slide images. So as we said, we have whole slide images, and this represents the physical slide into a digital format. And we are going to store it in approach like a pyramidal approach. So we need to know what data type will the whole slide image be stored on the device. There are different data types. The first one is standard image formats such as TIFF, JPG, JPEG, and so on. These formats support high quality image compression and are widely compatible with image viewing and analysis software. So you can basically open a JPG image on your phone screen or any traditional software, while the second one is specific, which is called vendor specific format. So many whole slide scanners generate specific file formats specific to the manufacturer, such as SVS or NDPI, and so on. The most common used one is SVS or Slide Virtual Slide, and these formats may offer additional features or metadata specific to the scanning technology, but it will require a specific software to view and analyze the whole slide image. So here you can download any whole slide image or get a whole slide image in a JPG or TIFF format, or any specific type such as SVS. So if you don't have the ability to get a whole slide image or go to an archiving place or a hospital to rent or get these whole slide images, there are different resources on the internet. The most commonly used one is the Cancer Image Archive, TCIA. It provides an extensive collection of cancer-related images where you can navigate to a specific region or a specific cancer and download the whole slide image. So how to do that? First, you navigate to cancerimagingarchive.net. It will open that site. If you scroll down, you will find submit your data if you have any data to submit, and access the data. Click on it. So you will have different options. One of them, if you are dealing with radiology, you search for radiology data, and the other is search histopathology. You click on it. So you will have different options to select from. The first is cancer location. So for example, let's select breast cancer, and for the data format you will have different options such as PMP, NDPI, PNG, and SVS. So let's select SVS, and you will have also, if you are focusing on a specific cancer type, you are selecting from here. And also for the staining protocol, we said we are going to deal with H&E or hematoxylin and eosine stain, so you click on it. So as you see here, the cancer location is breast, the data format is SVS, and the staining protocol is H&E. If you are not fine with each of them, you can cancel each of them by pressing on the cross, or if you need to make a new filter, you press on clear all filters. So by making a filtration to the data, you will find here 4 projects or 4 collections. As we selected, the location is the breast cancer, image a whole slide, and the stain is H&E. For each of them, you will find the species. They are related to human. You can find for any other animal also. So for example, let's open the first one. You click on view, and depending on the collection, it may open a view like that or a different view, but they share that you can view the whole slide images in this collection. As you see here, there are different whole slide images. Let's select the third one for case 4, click on it. It will start to load the image. This is the scanned or digital format. You can zoom in and zoom out. You can change the zooming from here, and the more you zoom, the more details you get. As you see here, you are seeing the cytoplasm and nuclei and so on. And if you check this number, you are getting 40x. You can navigate directly to 20x. It will be like that, and to 10x, and to 1x to get the abstract view like that. Let's clear our filters and try another search. Let's take a second example. For example, let's select the colon cancer and don't filter any other things. Just keep the data formats for all of them, and the same for staining protocol. And if you scroll down, you will find the location is the colon, the species is human, and you have different data formats, SVS, TIFF, and different staining. So let's select the third one, click on view, and select any of them. As you see here, this is the whole slide image of a colon of a human. You can zoom in and zoom out to get fine and detailed view, similar to the previous example. So at this point, you knew now what is the meaning of digital histopathology, and there are scanners that convert the physical to digital format, and the scanners have different parameters, and the scanners differ in the way they store the whole slide image. It can be general, such as JPG, or specific, such as SVS, and based on the storage, they have different approaches, such as tile format or pyramidal. And we saw 2 examples of getting whole slide images for research or personal purposes. And through the website, you are able to zoom in, zoom out. You can also apply some annotation, apply some processing on the data. But if you are going to work offline, you will need a software application on your device. So several applications are available for opening, annotating, and manipulating the whole slide image in digital pathology. The first one is a Piro Image Scope. The second is QPath. The third is Orbit Image Analysis. And we are going to work on each of them, download the whole slide image and import it in each of these applications, and getting familiar with different options in each of them. So Image Scope, it offers annotation, measurement tools. It allows also annotation on region of interest on any portion of the image. You can add text to draw shapes and perform measurement directly on the slide. QPath also can do that, can do annotating regions, draw shapes, add text, and perform quantitative analysis on the whole slide images. The third one, OIA or Orbit Image Analysis. You can also add markers or annotation, perform quantitative analysis. You can also apply machine learning based algorithms on the whole slide images. So this is considered a valuable tool for researchers and pathologists in digital pathology.